everyone. Good morning, good evening, good afternoon from wherever you're watching this year's SBI2 2020 7th annual meeting. We're very grateful for you coming and spending your time with us. I'm very honored to introduce our first keynote speaker. Lina Nilsson is a VP of Data Science at Recursion, which is a biotech that is reimagining drug discovery using machine learning. She was previously a COO of Enlitic and an innovation director at UC Berkeley. Her writings on open science, social impact technologies, and gender and tech have been published in the New York Times, Washington Post, and Science. She has a PhD in biomedical engineering from ETH Zurich, and she's going to be talking to us about some of the technologies that Recursion has been building to help turn images into the answers to questions and the cures for diseases. With that, we're all very excited to watch Lina's talk, so take it away, Lina.
Hello, everyone. I'm excited to be here virtually to share a bit about Recursion Pharmaceuticals and how we put machine learning models into large-scale production in order to find new medicines. I am going to be sharing three things in this talk. First off, I'm going to let you peek under the hood to see how our technology platform works, our machine learning and robotics. Second, I'm going to share a couple of case studies on how this plays out in practice for specific diseases, including for COVID-19. And then three, I'm hopefully going to have time at the end to wrap up by sharing our vision for the future of drug discovery and what we really mean by our mission to decode biology to radically improve lives. So I'm using Recursion, if you will, as a case study for how machine learning can be applied in industry. I want to just share a little bit about the company to help frame the story. We are based in Salt Lake City, Utah. We have nearly 200 employees actively hiring, so please reach out. And we've raised over $200 million to prove out our vision for how to fundamentally reimagine drug discovery to be more efficient. We've used the technology platform I'm about to tell you about to look across some 500 different disease models. That's really broad in comparison. A biotech of our size might more normally focus on one or a small portfolio of diseases. And this is really the main story of what I'm going to tell you today, how Recursion has accumulated one of the largest biological imaging data sets in the world, and of course, how we use this data set. Our core imaging data set is over 4.5 petabytes of data. That is 4.5 million gigabytes. But what we're doing is obviously not just collecting data points, an even more instructive mental model to think about might be that we are trying to industrialize drug discovery. That is, we're creating standardized large-scale production processes where we run the same assay week after week at massive scale while iterating through on what we're testing. So large numbers of potential drugs, compounds is what we call them, and also large numbers of very diverse diseases. It's popular, I think, to compare these kinds of mental models, like industrialized drug discovery, with what's happening in the big social media tech companies. Arguably, they're some of the original leaders in digital flexible industrialization. So for example, every day Google search capability infrastructure, that same infrastructure enables nearly 6 billion personalized queries, but Facebook, their core infrastructure enables nearly 5 billion unique personal updates to be shared by users. In some 10,000-foot view, what we're doing is similar. We're building one standardized platform that allow us to probe at an infinite, for practical purposes, number of different potential drugs and potential diseases using that same core platform technology. And another way to look at this, just since this is a longer talking keynote to give you some sense of how we think about this problem. Another way is to look back in time to be inspired. So Recursion is about drug discovery, But if you go back in time and you look at not the discovery of drugs, but the physical preparation of drugs, you can see a revolution that's already happened well over 200 years ago, 150 years ago in the pharmaceutical industry, where there was an inflection point where the physical preparation of drugs went from being something that was done within local pharmacies when a patient came in on an individual per patient on demand basis, and then transformed into an industrial process of automation, laboratory-based industrialization. And the results from that transformation was just a massive increase in scale and quality control, and ultimately in the ability for people like you and I to have access to high quality drug treatments. And that was really maybe the dawn of the modern pharmaceutical industry and what we think of as medicines today. And today we're looking at companies like Recursion and others creating a similar inflection points in how we don't just manufacture drugs, but in rethinking how drugs are discovered. And why do we need to industrialize drug discovery? Well, simply put, because approval rates of new drugs has been stagnant for nearly 40 years, and R&D costs at the same time has gone up tenfold. So in essence, the reason for this is in part because biology is really complex, and we understand very little of it. So as an industry, we need to find fundamentally new ways for discovering new medicine. The traditional approach to drug discovery might be to start with like a hypothesis of the mechanism of a specific disease and use this hypothesis to test for new promising drug candidates. That obviously works, but it's slow, and much of the, let's say, lower-hanging fruit has probably already been found. So enter, the 2000s and the increasing capabilities of new omics approaches, so like genomics, proteomics, phenomics, which are being designed in ways that can open new paths forward. And I'm going to tell you today specifically about a phenomics platform approach that Recursion uses that is hypothesis-free. That means it does not require us to have a theory ahead of time about what pathways or specific proteins are important in a given disease. But instead what we do is phenoprinting, that is machine learning analysis on human cell images using standard set disease-agnostic fluorescent probes. Why is this powerful? It's powerful because it's content-rich. We can capture thousands of features from each image that describe the state of those cells. It's scalable. That means that the same platform and analysis approach can be used across many programs. It's fast. In less than 2 weeks, as I will show you later on in this presentation, we can onboard a whole new area of disease. It's cheap. For $10,000, you can run by more than 100,000 phenoprinting imaging samples, or just 2 or 3 proteomics examples, or maybe 50 transcriptomics examples or samples. And then finally, it's machine learning compatible. On one hand, by this I mean that it takes advantage of lots of recent advances in data science, right, around image analytics, types of convolutional neural nets, variational autoencoders, multitask networks, and so on, techniques that have proven very powerful on images. But maybe more interesting and unique is that by having a single scalable, highly controlled assay, we can also physically add in various controls to our experiments that will allow us to do the most powerful machine learning possible. So a symbiosis between the generation of data and the analysis using machine learning. So let me take a moment to remind you of the punchline of this whole talk. It works. We have 4 clinical stage programs. That means 4 different disease areas we are ready to test potential drugs in human patients. And with that, let's start to learn about the actual machine learning and the robotics automation. Much of the machine learning we do at Recursion is a form called representation learning. That means that rather than building classification models that focus on a set of predetermined outcomes, like specific diseases, we build high-dimensional embedding spaces, so mathematical descriptions, numeric descriptions, high-dimensional vectors that can describe cells effectively across a large range of possible cell states. So a classification model, right, might be, am I looking at disease A or disease B? Right? Is this a cat or is this a dog? That's not what we're doing. We're providing, creating these high-dimensional descriptors that are upfront agnostic to the meaning of those biological states. So this here is a very simplified version of an embedding space, just for the purposes of this presentation, where we've taken 3 dimensions, something the human mind can handle, instead of the over thousands dimensions in the original analysis. The red dots you see are various diseases we tested on our platform, and the blue dots represent potential drugs that we have tested against these diseases. In this example, compare the red dot of a disease with a green dot of healthy cells, and what they would look like in a high-dimensional space. And we can see some drugs stand out as being promising of, when added to diseased cells, causing those cells to begin to look something like a healthy set of cells should. Various types of this form of high-dimensional analysis allow us to understand what's happening to the cells in our phenoprinting assay in a way where each cell state it's unique, but it's also giving us a complex, nuanced information in a way that's quantifiable. And we can then use this phenoprint assay and these high-dimensional embeddings to do a number of things. I can give a couple of examples of what those things are. Actually, I guess I need to step back for a second. Here, I'm just showing some images of microscopy cells from our assay. Just showing what I just described with words with some images, where we're turning these microscopy images of healthy cells, of diseased cells, plus drugs into these high-dimensional numeric descriptors. And with this, what we can do is, first off, of course, find potential compound hits. So that is drugs that rescue diseased cells so that they look healthy in our assay. In addition, and maybe really interesting and unique, is that we can also quantify side effects, all the things that these potential drugs do to the cells that we can pick up in the microscopy that have nothing to do with the rescue of the disease. And by and large, a lot of these are unwanted side effects. They might indicate problems in future downstream analysis with that potential drug. And then secondly, there's all kinds of more detailed analysis we can do to understand these potential drugs better. So use these assays, these representations, to begin to predict toxicity in downstream analysis that would be done, or understanding the mechanism of action, not just that the drug works, but why it works. And then finally, from the example list here, I will talk at the end of this talk a little bit about how we can use these images to begin to build biological networks that will let us make new types of insights from what Recursion has done in the past. Here's just an example with some real data from a disease called hereditary hemorrhagic telangiectasia. And you see some of the images here of us adding different amounts of drug X, a compound we tested on our platform that rescues this disease, HHT. And on the right, you see just one example of how we take this high-dimensional data and compress it in various ways that allow for easier human analysis, in this case into an on-target an off-target score like I described on the previous slide. So that's essentially collapsing this high-dimensional space into just the vector here and an off-target score that captures the rest of the insight. And this right here is just another way to look at the data I just showed you, or the examples I just showed you conceptually with the video in the last few slides. This may be a way that some people in the audience are more familiar with. We have a deep learning architecture that we use to extract deep learning features, so these descriptors of the original high-resolution microscopy image across a number of different channels of fluorescent probes. And then we take these features, and in the examples from the previous slide for the disease HHT, we deconvolute that into the part of that set of features that represent the on-perturbation component. That is the component that can take you from something that looks like a disease cell to something that looks like healthy cells. And then an undesired component of that feature signal. So that's all the other changes that a potential drug might induce in the images that is not related to reverting the effect of the disease. And here's one more example of real data for this. This example looking at the growth factor TGF beta. And the take-home here is just to reinforce the point I'm making about the value of representation learning as a machine learning modeling approach, particularly when you want to do what we're doing of building a reusable platform that can be applied to many problems of the same type. In this case, the different problems for different diseases that we care about. So TGF beta is a primary driver of fibrosis in a number of debilitating diseases. So, for example, in pathological wound healing, it's essentially tissue scarring in places or in ways that are harmful for the body. So what you see here on the left is, again, the high-dimensional platform results collapsed into just 2D for the purposes of this presentation, where you can see one example of a compound, it's REC 01049737, that rescued deceased cells into something, looking like healthy cells. And then in B, you see us running a traditional low throughput confirmation assay for an output collagen, which is an output related to fibrosis and scar tissue formation, and just confirming that the results of what we see in our novel phenomic assay can also be capitulated, in this case, by this more targeted low throughput assay. And then similar in this second example here on the right, we're now taking that second step that I described of not just finding hits, but understanding why they're hits. And in this example, simply saying, in this high-dimensional space, how does the compound we cared about, this hit we found, how does it compare to lots of other compounds that we have that we know how they work? And in this case, we see that our compounds looks a lot in the high-dimensional space like a group of compounds that affect a protein and a pathway called ALK5. And again, you can confirm that this is indeed the activity of this compound, also with a more targeted assay, specifically looking at ALK5. So in A and C, we're taking our high throughput output agnostic assay and getting the results that are otherwise only attainable if you already knew ahead of time which of many thousands of potential target-specific assays you were going to look at. Just to highlight one of the benefits of the agnostic phenomic assay. And how do we in practice run all of these biology experiments for machine learning? We have a huge robotics laboratory where we run up to 850,000 wells per week That's several million microscopy images going through on that weekly basis. And again, 4.5 petabytes of data. How much data is that really? 4.5 petabytes of data. If you took that quantity and you turned it into standard high definition movie and you wanted to watch it from start to the end, you would have to watch that movie for 24 hours a day, 7 days a week, 52 weeks a year for 200 years. It's a lot of data. And that brings us really to one of the core points of the talk. Your machine learning model is not worth much if you can't put it into production. And here I'm showing you a much simplified snapshot of the digital part of our pipeline. So what happens when the robotics are done? I'm just highlighting a few key areas. For example, being able to handle image uploading at a speed and volume per second that is tracking the capabilities of companies like Twitter, or for our weekly feature extraction, creating these high-dimensional embeddings. We're running the equivalent of nearly 20 years of single desktop computing just in that week, and then following that by analysis and interpretation of more than 600 million human cells a week. So it's clearly pretty simplistic to think about what we're doing is just applying machine learning to images. There's just massive machinery that has to sit around these machine learning models to ensure that we have high performance, that we have quality, that the insights have relevance. And so to take just one of the components here from this list on this slide, if we look at, say, image quality, we, for example, use data science method to make sure that our images are in focus, so that any image that goes to our downstream representation learning models are high quality images. Or let's see, consistency, execution error. We use machine learning and data science methods to make sure that all of our reagents, all the inputs into this robotics pipeline look the same this week as they did last week and as they did a year before. So that's another place where we use machine learning and data science to ensure that our processes are high quality. And like I mentioned briefly before, this is not all digital. It's also physical control systems. So for example, controls that we embed into our experiment to help our machine learning embeddings and models. So that's actual physical additions to some experiments that help us build the best models possible. One example of this from the previous slide, just to dig in a little bit more, is around building predictive models. I mentioned this really briefly, being able to predict what's going to happen in downstream assays, such as toxicity. So here's an example for a specific toxicity assay called HERG that measures the risk of a compound, of a drug, causing cardiotoxicity, toxicity to the heart, if it was eventually to be given to patients. And here, what I'm showing here with this enrichment cycle is that when Recursion collects its very large data sets to be able to build these types of predictive models, we also run models during the early stages of creating the data sets in order to make sure that we create data sets that have lots of the kinds of example that we want to see in order to train our final predictive, in this case, cardiotoxicity model. So in order to do all of this in high quality, at scale, we're embedding various types of machine learning models throughout the process of creating our new industrialized systems, in this example, in order to create training sets. And on the right, you simply see a chart showing how both the hit rate, the number of compounds in the positive class, is enriched with this kind of strategy, and how that then eventually, as the next step, causes us to have higher model performance because the models have had larger and more diverse set of positive examples to look at during the training of the machine learning model. And then at the end of this big phenomics platform of these terabytes of data coming through every week, we need to be able to show all of this data, of course, to our biologists, to our chemists, and to our other drug discovery team experts. And so another take home is that it's not enough to have the machine learning models. It's not enough to have the pipelines. You also have to have tools at the end to capture all of this data and all of this complexity, while at the same time having those tools be easy and quick to use. So this, I think, is a critical area as well that maybe often is skipped over in public discussions about how AI algorithms, how machine learning can be really used in practice in industry. How do we take all of these high-dimensional embeddings and turn those into a series of tools that allow our human experts to focus on the most important data points out of all of the ones generated every week. And then, since the name of the company is Recursion, as part of telling you about how our technical platform works, I wouldn't want to miss explaining at least one way that our industrialized drug discovery process is recursive. That is, it is the repeated application of the procedure to successive sets of results. So here's that example. We take the output of our phenomic platform, and we can search over 1 billion, and I think this number is going up very rapidly, but 1 billion for this slide at least. It's the latest I have updated, of commercially available compounds from various vendors, various sources outside of Recursion, so that we can very rapidly find the best next set to test. And then we can iteratively go through this process, run the platform, use our large-scale search capabilities to find the next set best of compounds, and then rinse and repeat for several rounds. Of course, an important part of this is like how you do the search. So here again, we can combine this with both sort of industry best practices types of search algorithms, in addition to our own in-house tools for finding compounds that we think are going to be promising for the next round of screening. And maybe one other thing that's interesting about this slide for people who care about this process of how do you really use machine learning in practice in real company settings is that it also shows how our phenomic screening, this technical platform that I've been talking about today, it doesn't sit in isolation, right? But it integrates into what you might call a full stack of drug discovery. That means with bench validation, with in vivo animal studies, and eventually somewhere on the right off of this slide, with human trials. How is this platform affecting the trajectory of Recursion? This is a quick just summary of the scale that this platform has grown over since Recursion started, from very small scale semi-manual process back just 4 years ago to a fundamentally different level of scale today in 2020. And I don't even know if these numbers are completely up to date. So one take home here is, again, the industrialization of the process and how rapidly this kind of mindset to the problem allows you to scale quickly. But second, in blue, you also see how we don't need to scale the human input that we're putting into this process in the same way. So in 2019, even as we reached 300,000 of these mini experiments per week, we still only had 2 human expert designers for all of those experiments. And today in 2020, we don't even have full-time people dedicated to this process. So that kind of automation of this core technical platform means that we can dedicate our human resources, our brains, to both pushing on the next frontiers of what this platform can do, the new types of predictions, the new types of insights, as well as on downstream analysis just on the most promising compounds and the follow-up analysis, either on the biology bench or in vivo trials. So you don't have to take my word for all of this. If you want to pressure test some of what I've told you yourself, you can. We have opened up 3 gigantic data sets from our phenomics platform to the world. And all you have to go is to go to rx.ai to download them. There is a gigantic set called RX1 that's optimized for understanding how you can relate and compare noisy biological data from many different batches, so for across long periods of time, because that data set was collected over many months. We have RX2, where we've released both images for immune perturbations, for something very relevant to drug discovery, and also released our actual deep learning embeddings so that you can play with this data set even if you are not a machine learning scientist and able to create your own favorite embeddings and model outputs. And then finally, there's RX19, where we released both images and deep learning embeddings for our work on COVID-19. So I invite you to join and to explore all of these gigantic data sets yourselves. Next, I'm going to transition from talking about the platform itself to give you these 2 case studies, starting with C. difficile, bacterium a lot of you have heard about for its role in various bowel diseases. So here are some results for C. diff in human cells on our phenomics platform. And again, C. diff is involved in colitis, irritable bowel syndrome, and so on. And part of what's really neat here, and that's demonstrating with these slides, is again that the platform is agnostic to how the drug compound works. So in the red line on the left is a compound that targets not the bacterium, but the host, so the human. It builds up your defenses against this bacterium. And in green is a compound that we also found on the platform that instead directly targets the bacterium, which will be the more common way for a drug to work against an infectious disease. And then as you can see on the right, we can take this platform we built and rapidly turn it around to test a new disease model. So here we tried a type of E. coli to see if the same compounds would work in this somewhat related disease model, and we see that the host targeting compound, the one that protects the human, also protects against this second platform. So it essentially allows us a way to both differentiate between different compounds very rapidly using the same assay, and also to start from the starting point of one specific disease to look at other diseases related to it where the same compounds might also work. So rapidly jump essentially from assay to assay because of how this whole platform is set up. And just this is really rapidly, quickly just showing you that we can confirm these host versus bacterium mechanisms in more targeted, less scalable bench science assays. So here's just one of many examples of that, where you can see that lack of human cell membrane integrity, one of the hallmarks of the diseases caused by C. difficile, is rescued by the addition of the compound that we found in the first big phenotypic screen. And I don't show the data here, but we also advanced this particular compound scaffold into in vivo animal models, and it works there as well for C. difficile. So for example, typically you might see animals in the standard animal assay we use succumb to death after 2 days in the disease model. But with our molecule, we've increased that survival rate at day 2 by a significant amount. here is a third, and I think final example of how the phenomics platform can help you compared to a traditional biochemical targeted assays. In this case, help you to avoid false positives. The x-axis here on the right are the results in the phenomics assay. You can see most of that text. And on the y-axis is a standard single endpoint biochemical assay. And the red circle shows a compound that worked really well in the single endpoint assay, but did not work in a cell assay. So if you had done the simpler, the more narrow single target assay, you would have gotten a false positive. And likely the reason that the phenomics platform gave you a more nuanced picture here is partly it partly could be that this compound is not very cell permeable, so it wouldn't be picked up. But in other times, we also see compounds that they work well in that isolated biochemical assay, but in the full cell context, they might bind to any one of the other 20,000 proteins in the cell, rather than the specific target you might have in mind from your single endpoint reductionist biochemical assay, among many other problems that might not be picked up in the biochemical assay. And then, of course, each phenomic screen, as I briefly mentioned, can anchor it into a detailed research operating plan. And this is very high level of what that looks like for C. difficile. So that is anchoring the primary screen, the machine learning analysis, both with sometimes more detailed secondary assays, such as the cell junction barrier function assay, all the way down through a compound that eventually is ready to be tested in human trials. And so what we're doing today, even though I've focused on this core phenoprint screen and how machine learning, robotics, and other new technology tools are being used there, that's the focus of this talk, but we're also applying that kind of mindset to that full process and this full pipeline that I'm showing here. And here, related to this story of how the platform really, in practice, gets applied to real-world problems. Do these machine learning models really let you do answer questions that are relevant in the real world? The platform is really robust and very reproducible. So this is just showing 3 compounds, 3 potential drugs that work in our core assay, and how those results repeat and reproduce week after week. This is really important because one of the core ways that drug discovery platforms like the one I've been showing here work, like how that initial finding of a compound that could become a future drug to help people works, is that chemists need to iterate on your initial chemistry starting points, just iteratively, to get to a chemistry that's really optimized to be your final drug. This process is called SAR. This is search for the structure, activity, relationships. And having a platform that's highly reproducible and precise is critical to allow you to do that kind of iterative chemistry where you're replacing parts of the original compound, as shown here with a core with ABC. And as you replace different parts, you want to be able to, in a quantitative way, see how the results in your assay change as you replace these different parts of the original compound, and see if you can find a compound that is even better. And this kind of iterative process is a cornerstone of many drug discovery programs across the industry, and being able to do it with a high throughput agnostic platform the way we're doing it is really, really powerful. And with that first case study, I'm going to transition to briefly tell you about the COVID pipeline that we have built this late spring. Big take home here is that in less than 4 weeks, we could go from this just being an idea in someone's brain to having results coming out from the platform, finding compounds that worked and that didn't work. And that's including all the logistics around setting it up. we've tested 1,500 compounds, a bit more, and done that both in 2 different cell types and again, shared these results both on Campbell and on rxrx.ai, as I shared before. So a lot of results, but I'm just gonna, in the interest of time, highlight a couple of drugs that you might have already heard of in the news from the 1,600 we tested. So we looked at drugs that already were in various stages of testing for coronavirus, COVID-19 or related diseases, or that have been approved for other diseases, so they're on the market and considered safe. So one you might have heard of is remdesivir, which did work in our assay. So it means that it was able to rescue deceased cells that were being exposed to COVID and or inhibit that infection and cause those cells to look like healthy cells. And as you may know, remdesivir, while at the time was just beginning to be talked for, it's now in various clinical trials, including by Gilead, and it's also given to patients in hospitals in the United States for treatment of COVID under an FDA emergency use authorization. So those are things that happen after we ran this test. Here's the second one that you might have also heard of, hydroxychloroquine, another drug that has been much talked about for COVID-19. In our hands, it showed some efficacy when we used a commonly deployed animal model, but at the cost of having very large unwanted side effects. So you can see that with this back hook in the left graph for hydroxychloroquine. But importantly, once we tested this compound in human cells, we actually didn't see any wanted effect, any rescue at all of the COVID infection of these cells. And all we saw was large off-target side effects. Secondly, in addition to infecting cells with COVID-19 virus, we also looked at another concern related to COVID. So this is cytokine storms. So this is the human immune system out of control in response to the viral infection. And where sometimes, as many of you are aware, I'm sure, the out of control human immune system could be a larger issue than the initial direct infection itself. And here's some graphs again showing how we first rapidly build various cytokine storm models and then tested a large array of already approved drugs in order to see if we can find some that have a positive effect of the disease states. And there's some interesting hits in there. And that brings me to my final few slides. Beyond these examples and the work that Recursion is doing today on specific disease programs, dozens of specific disease programs, where do we go from here? And what is really our vision for the future of industrialized drug discovery? To simplify a bit, the approach I have shown you up until now, it's one of brute force, right? Like we don't have a specific hypothesis, just test all the combinations, hundreds of thousands of compounds against hundreds of diseases, and just see what shakes out. But now we're beginning to move to the next phase. If you have this much data, you can take these massive data sets and begin to build out predictions and a map of human cellular biology. Essentially, begin to combine results from all the separate assays we run in order to reconstruct and discover cellular pathways and signaling relationships so that we can begin to decide ahead of time in an ever more clever way which experiments we should even run to begin with. Right? And so this slide shows just a tiny subset of genes that we've tested on our platform using CRISPR. So we compare the high-dimensional phenotypic signal, again, between various gene knockdowns and begin to take that to build a gene map of defect on cells on gene knockdown and gene signal. So in the heat map here, dark red means more similar and vice versa for blue. So just to like highlight one small example from what's actually a very large data set here, knocking out by CRISPR of KMT2D and KDM6A, two different genes that are both involved in the same human disease, Kabuki syndrome, look very, very similar in our platform, showing that for this very serious multi-system disorder, various genetic perturbation that lead to that same disease look the same. And you can imagine beginning to build up all sorts of powerful insights about how diseases are related, how potential drugs are related, how potential drugs and diseases are related a priori before the specific combinations are tested in our platform. And this is showing that same concepts, now looking at compounds like potential drugs, where you can differentiate between different mechanistic targets and also see nuanced subclustering within a target class. So here, for example, for mTOR inhibitors, where different compounds might have different potency, so work at different concentrations to work, or have different polypharmacology, where they're also hitting other targets in addition to mTOR. And building these large-scale maps by testing many thousands of different compounds of different genes and so on, we can begin to tease out this kind of nuance. And then eventually bringing all of these things together, where we're building out ever larger combined comparison maps of human cells. So that one day we will be able to predict things ahead of time, such as which compounds are likely to work in which disease, or given disease X that I care about, how do I design the most promising initial compound chemistries? And that's really, I think, ultimately the vision of where Recursion wants to take machine learning for drug discovery. We want to use it to decode biology, to build these large-scale maps in order to radically improve human lives. And with that, I'm ready to wrap up. I have shown you a bit about how we're industrializing drug discovery using machine learning and automation to recursively run the same assay again and again and apply it to many diseases. With this approach, right, we can transition between these diseases really rapidly and bring on new diseases really quickly, like I showed you with the COVID example and with C. difficile for irritable bowel syndrome. And then finally, Recursion is now using this massive scale to begin to build a map of human cellular biology. And with that, I say thank you, and I invite you for questions.
Thank you for that talk, Lina. That was fantastic. We have some great questions here from the audience. I'm going to take the liberty to paraphrase a little bit. But let me start by just asking, do you use specific tissue or cell types that are physiologically relevant? Or does any cell type essentially image pretty well? And do you have preferences for 2D versus 3D?
Right. So we have the capability. We have run dozens of different cell types. We work with patient cells also in addition to that. So that's a broad span. But we try to focus in on certain sets of cell types that are proven promising in our hands so that we can really scale and run really rapidly for the big screens. So there's definitely a strategy around not trying to find the perfect cell type for every single application, but really being able to run the initial screening step at massive scale, which can be hard to do with certain cell types. I presented before around the 3D questions. on some work we've done in organoids, so for example, beating heart cell organoids and spheroids. And that's something we've done at kind of a medium scale at Recursion, and we're now looking to do it at much larger scale. So if you're interested and you're looking for a job, reach out. So we're exploring a number of different techniques to get to that increased physiological relevance.
And how transferable do you find some of these results are across cell types? Do you find that generally if you've discovered a phenotype in one, you find it in others?
It all depends on the pathway biology of the disease. So some things show up in a lot of our different cell types, and some things are just more specific, right, to an immune cell or neuronal cell, et cetera. So it's a kind of hard question to generalize on.
Yeah. Can you talk a little bit more about sort of the biological interpretation of the embeddings? I mean, you've shown in your talk there that there are some ways where you can sort of try to figure from what's around it, but how easily can you then turn an embedding into what a biologist recognizes as a phenotype?
Right. So I guess the question here is like having, for example, features that are human interpretable, like the shape and size of the nucleus, the number of mitochondria, et cetera. And early on, we did work at Recursion trying to match features this way. And we've kind of abandoned that as a strategy, basically saying if we truly want to be target agnostics, we truly want to pick up all of the signal from the cell, it's self-defeating for our approach and our strategy to try to do that mapping. Because if we do that, we're leaving so much on the table just because it doesn't perfectly match to something that we can think about like in a human interpretable way. And instead, we leave that human interpretability part for later orthogonal assays where we're trying to elucidate the mechanism in more detail. And that I think is the more powerful way to really leverage these new deep learning techniques, right?
Mm-hmm. Why the preference for high dimensional embedding? Do you have a sense of the effective dimensionality of the biology of interest in any of these cases?
Yeah, this is a really interesting question. I'm interested in this too. Being high dimensional, kind of going to the last question, right, allows us to be totally agnostic about what's important and what isn't, right? Like, for example, we have had mitochondrial diseases where the signal does not map to the mitochondria specifically or solely. And then those potential drugs that we've found have validated in later assays. So I think it can narrow what you're looking at to get too narrow about what are the dimensions or the features, the things you care about in the cell. So we really want to get away from that. And the kind of biology we've been looking at to date, we do have kind of too many dimensions, if you will, like we have too many channels. We are allowing the initial featurizations to span more dimensions than we really need. And then we narrow down to what is actually important for that particular disease to determine, to differentiate between the healthy and the disease state. But that's definitely an interesting question for us, right? Because when you're running things at these massive scales, doing that extra bit of work that might not matter if you're running 1 plate or 100 plates really starts to add up in logistics. So we're always, for sure, into the future looking at ways that maybe we can kind of just make sure that we're hitting that balance between capturing enough to capture states that we might not have anticipated, but not kind of going so overboard that it costs us time and money and frankly could lead to less signal than we want and more noise.
Mm-hmm. How do you incorporate feedback from the in vivo studies on a drug? So once you get further down the pipeline, how easy is it to incorporate stuff backward?
Yeah, thanks for that question. Yeah, so we run lots of animal studies also on the most promising hits. After we've done bench science validation, that's the next stage. And as I mentioned in the talk, there are 4 compounds that are now also ready for human clinical trials or in human clinical trials, depending on. And so we don't today feedback the specific information from the in vivo animal studies. It's a problem of N, right? Of what are you optimizing for? And if you have the number there that you can have is much smaller than what we can have on the big platform, right? And so we don't want to skew ourselves too much for that very small test set. So the 2 things, or 2 of the things that we've done instead to date are 1, testing compounds that are in the clinic or have been in the clinic on our platform. So We've tested hundreds, and I haven't talked about the details of this publicly before, so I can't really get in the weeds about it, but we've tested hundreds of compounds that are available on the market on our platform to confirm that the platform can do what we think it can, and use that to help improve and iterate on the deep learning. Another thing that we do today is run very large, massive experiments simply for this map of biology, right? Then again, lets us improve the general deep learning algorithms to distinguish between biological states better. And then the third thing, I guess I should say, and is that we have bought a company called Biome a while back. That's public information too, and that is an in vivo research company that uses machine learning techniques at the in vivo stage, and we hope will have some ability for us to kind of tackle that part in a way that it kind of works with the way Recursion operates, without letting us hang on to very small numbers of in vivo results.
Question that just came in that I think is really interesting. You highlighted the importance of consistency, reagents, images, things like that. How do you balance keeping consistency with embracing new technology?
Oh, interesting. Yeah. Through fierce debate. Yeah, this is really tricky, right? And I mean, maybe this is like a human kind of like how you work together question. So we divide up our work into things that are divided into like the maintenance that we already have, the building off of that specific work, and then a whole area of bold bets within Recursion, where there are groups of people just focused on rethinking how fundamentally how we do things. Like there was the questions about the mathematical space we used around cosine similarity I saw, and like that's not the only way to do it, but the cost of switching, obviously, in a pipeline like this is really high. So we have separate teams whose job it is to say, what could this look like in a year, in 5 years, and let's build that out. And then take the most promising things and integrate that into the main platform. But it's tricky for sure.
Yeah, I can only imagine. Time just for one or two more questions. One question that I had personally is the release of all of those public data sets. What benefit do you hope those will have for the community? And is there any benefit that you hope that specifically those will give to Recursion, the release of those public data sets? Because putting things together like that is quite a bit of work. Yeah, for sure. I mean, I think that the machine learning community is really good about sharing and the field that's really been spurred by that kind of attitude of putting both, not just results, but also tools out, like Google's TensorFlow, et cetera. There's lots of examples of that. It's hard as a small company to contribute to that and to balance like you growing and doing that work, and putting out these data sets is, I think, a great way for us to give back to the community and be part of that kind of a mindset and way of operating. So, I mean, fundamentally, it's just like it feels good, and we want more people to be working on these problems. But then, I mean, for Recursion, we're hoping that people see those data sets and decide they want to come and work with us and play with the other 99.9% of the data. And we have had that. And then, of course, it's a way for us to show that what we're doing is real, right? Mm-hmm. Yeah, there are really some phenomenal resources. And the last question I'll go ahead and ask is, do you have a sense of the range of mechanisms that can't be detected by the platform? What are the limitations that you guys are working to overcome, to the degree you can talk about it? Yeah, ask in a couple of months. So we are doing great, big research work on this in-house, running large experiments to try to get a better sense of this. Where is morphological profiling going to be the most promising, and can we use those insights to guide what kind of diseases we work on? And then in addition, what are the complementary methods that we want to scale further at Recursion to capture the broadest biology possibly? Yeah. But I think it's both kind of an intrinsic question about morphological signals, but there's also something here, right, about continuing to improve deep learning models, continuing to improve the process of actually running the experiments. I don't think we or anybody has really reached the limits of what can be seen in morphological imaging, right? So I think there's a lot more
We just improve methods to get at. So I guess there's a question of what can we see today, and there's a question of what do we need to do to capture even more.
I think that's a fantastic note to end on. Thank you so much for your time. Thank you everyone for your attendance. We really appreciate all the great questions. Please enjoy the rest of the SBI2 meeting.